เป็นภาษาอังกฤษนะคะ Good morning, ladies and gentlemen. I'm very delighted to be here to present about the antimicrobial resistance, genes, and survivability characterizations of Salmonella using whole genome sequencing and genome-wide association study. My presentation is covered 4 parts. First, I will talk about the introduction just briefly, and then I will move on to material and methods. And after that, we will consider results and discussion together. And finally, I will show you about conclusion. Yeah, Salmonella is an important zoonotic foodborne pathogen which is recognized as a major public health concern. Food animals are the major growing cause of salmonellosis, especially in pig. But not only salmonellosis have a problem in nowadays, but also antimicrobial resistant Salmonella is increasingly serious problem to global public health concerns, including Thailand. Moreover, Salmonella can be persisted in the swine industry. In my previous study, we found some persistent strain and cross-contamination along the swine production chain. So I got the question that how can Salmonella survive in the environment? So it might be biofilm formation or anything else we don't know yet. All the problems we can figure out by using the whole genome sequencing and bacterial genome-wide association study, or GWAS. However, whole genome sequencing is the new technological advancement which offers a fast way to sequence bacterial genome and is increasingly replacing other molecular genotyping. And it can be used for outbreak transmission investigation, genetic analysis, microbial identification, predictions of antimicrobial resistance, and anything else. But this is the work for whole genome sequencing that you have to do the DNA extraction first and prepare the library that you have to make a short fragment of the genomic DNA and then you append with the sample index and the adapter and put them together in the sequencers and interpretation. How about bacterial GWAS or bacterial genome-wide association study? It's a brand new bacteriology to help deepen the understanding of the genetic variation associated with the interested phenotype, and it's a new research opportunity that is driving forward by advances in genome sequencing technology. For the objective of my study were to detect antimicrobial resistance and to investigate the survivabilities of Salmonella species in swine industry in Chiang Mai and Lamphun, Thailand province, Thailand. Chiang Mai and Lamphun province, Thailand. $124 Salmonella strains were randomly collected from farm, slaughterhouse, and marketplace in the Chiang Mai and Lamphun province, Thailand, and do the Salmonella isolation and extraction. For this, we do the extractions and do the whole genome sequencing to test for the antimicrobial resistant genes. And we can get the whole genome sequencing result to do the phylogenetic tree. The same isolate will do the test for the biofilm formations to be asked the phenotypic difference and map them with the high and low biofilm formation and do the genome-wide association study. For the GWAS, I will explain you more clearly in the other next slides, but for the next slide, I will just explain how to do the antimicrobial resistant gene testing. Antimicrobial resistant gene testing uses a tool-informed comprehensive antibiotic research database or CARD. You can go to this website and then click on download. And you will get like a FASTA files that include all the public antimicrobial resistant genes. And pop them in the big database and click submit, and you get the show number 1 or pink one is mean that is 100% similarity. Further results and discussions of antimicrobial resistant gene testing. So these are the results that I get, but it's hard to explain. I will explain you more clearly in the next slides. So resistant genes confer to many kinds of the antimicrobial resistant genes, as you can see. With the tetracyclines, ESBL, and aminoglycosides resistant genes were found with a high prevalence in the swine industry. Moreover, I found the outer membrane effect glyprotein quaternary amino compound that is involved with the antiseptic and disinfectant with like quite high 25%. And they have the relationship between resistant phenotype and resistant phenotype. And the sensitivity and specificity of CART that predict resistant phenotype to express phenotype here. CART is good, convenient, and fastest tool for analysis antimicrobial resistant genes. As you can see, tetracycline, aminoglycosides, penicillin, and cephalosporin are not able to be the drug of choice to treat the salmonellosis in the future. Sensitivity and specificity that calculated from the outcome will provide a good concordance between resistant phenotype and resistant genotype. As you can see, many strains harbor antiseptic and disinfectant resistant genes. This might be the one reason why Salmonella is very difficult to eradicate from the swine industry. So that make me study about the survivability. Before I show you the result of the survivability, I would like to show you how to do GWAS. So this is the whole genome sequencing tree that is labeling low and high biofilm formations. Low biofilm formations is green and high biofilm formations is red. And then I just select the isolate that have the association between high and low biofilm formation, and the total isolate is 50. And I did the GWAS by using 30 base pair searching based on the biofilm formation. And the genes that hit is 30 genes associated with the survival ability of Salmonella in the swine industry. So this is our result. They have many genes that are associated with the survivabilities. They have metal uptake, antimicrobial resistant genes, antiseptic resistance, or any straight factors. So, as you know that having raw meat or non-cooked meat should be avoided. And using antimicrobial resistance should be controlled by a veterinarian, dentist, doctor, and pharmacy. And Salmonella can survive in the swine industry, Thailand, with many kinds of processes. So cleaning program in the swine production chains should be improved. Okay, thank you for your generous attention.